Welcome at the last day of the Life and Space conference organized by Polish Astrobiology Society. I hope you all had a great time during our sessions. I certainly did. And this is the last event of our conference when Lynn Rothschild is going to present her talk about synthetic biology in space. Lynn Rothschild is a PI at NASA Ames Research Center. She actually she did one of the first astrobiology conferences ever at NASA Ames Research Center. So she is a very recognizable figure, and I'm so grateful, Lynn, that you agreed to give us this talk and close this conference with a wonderful presentation. So the floor is yours, Lynn.
Thank you very much, Tomas. What Tomas failed to mention is he also spent several years in my lab, and I'm a big admirer and very grateful for what he's done. I actually ran the first 3 astrobiology science conferences, which is quite a job. So I know this has been quite an effort to put together, and I'm really grateful for anyone who's still listening in, and for the polls who are not watching the World Cup. I'm also very grateful that you're willing to hear my talk instead of watch football. So this may seem like a little bit of an odd talk for an astrobiology conference, but I just remind you that astrobiology can really be summarized by 3 sound bites. Where do we come from? In other words, where does life come from? Origin of life, all the way back to where do we get the building blocks for life, the prebiotic chemicals, to how do you get the prebiotic chemicals? How do you get habitable bodies? Arguably all the way back to the Big Bang. Then the second question is the flip question to that. What's going to happen in the future? And it's not just, are you going to get a chocolate cake for your birthday, or that, sort of silly thing. But we know that the sun is a star, and just because we're here, it doesn't mean it doesn't stop burning, and it doesn't stop getting brighter, and the chemical composition changes, and the habitable zone within our own solar system is going to change. And of course, the Milky Way and the Andromeda galaxy are heading for each other, et cetera, et cetera, et cetera. And, you can take a very long view, and what will that mean for us? So it's sort of astrobiology in the future. And the third question, which everyone loves, is the search for life elsewhere. Now, about 15 years ago or so, I was asked to start a program in synthetic biology for NASA. And I'm an evolutionary biologist, astrobiologist by training. And so this seemed a little odd. But I've also have it in my mind that synthetic biology is not just useful for human space exploration, which is true, and that is sort of part of this question of what is the future of life for astrobiology, but it also has a real role to play in astrobiology as we think about it, normally the search for life and where do we come from, as well as, again, where are we going? Most of this talk is going to be focused more on the future, getting humans off planet. But if you stick with me, I will get to the more sort of traditional astrobiology at the end and wrap it all up for you for 2022. This is my last talk of the year. Yay. I hope it is for many of you, and you can move on to holiday parties and all that sort of thing. So let's go from here. Let's see. There we go. Now, we're all on planet Earth, and it's very easy to communicate when you're on planet Earth. This is a picture of NASA Ames Research Center. My lab is way over to the left. What you're seeing is the hangar there where the Akron and the Macon were kept. It right now doesn't have its shell on it, but just for historical interest, that's also where we ran some of the first astrobiology science conferences, which got to be known as AbSciCon. But when you get off the surface of the Earth, and here's a Landsat image of the San Francisco Peninsula, and there's NASA Ames for you, communication becomes much more difficult. You have a little bit of a time delay. You can't necessarily see the person you're talking to. You can't get materials back and forth. And then as you go further into space, here's a wonderful Earthrise image, not from Apollo 8, but this one is from the Japanese Space Agency, JAXA. We realize that it's even more of a communication issue. Now, we're first talk that I've ever given while we have a lunar mission going. We don't have humans on Artemis 1, but we're heading that direction. And as we all are again painfully aware, it's several days to get to the moon and back. So you have to have a certain amount of independence. And as we go even further, we go to Mars, the time gap and the physical gap becomes even greater. Right now, we're talking about a 6-month delay from going from planet Earth to the surface of Mars. And just to really drive this point home, I don't know how dusty your screens are, but perhaps you can pick out where planet Earth is in this actual photograph. And here, I'm going to help you out. So there we are, this little dot that contains everyone you've ever known, all the history of planet Earth, whether it's geological, biological, human history, it's all wrapped in this little tiny dot. So clearly when people go to Mars, and I'm not saying if, I'm saying when, this is going to be a real issue. And just because I work at NASA and I get to do this kind of thing, here's a picture that the Cassini mission took around the Saturnian system. And once again, I ask if you can find planet Earth here. And again, I'll help you out. So even within our own solar system, we become a little tiny pale blue dot very, very quickly. And so as we do this, and we're very cognizant, we're very aware of this gap, we also take with us the needs for humans. Now, here's a picture I took in New York, and it's a great picture because it shows all the basic needs that we have for humans. We need transportation. We need life support, food, we need medicine, we need to recycle our waste, we need clothing. And I always laugh about the oxygen because people in the military say, well, we have it very tough too, going to extreme environments. And I say, yeah, but you get gravity and you get oxygen. We don't even get gravity and oxygen. So all these things that some of which we think about and some of which we don't, we need for life support. Of course, we need habitats for humans. We need power, we need heat, we need light. We need radiation protection, and this becomes increasingly important as we go off planet. We need forms of communication, again, stressing that we're going to have a time difference and a great physical distance. So we're going to have very good communication as we go forward. And just because this man's waving a wad of dollar bills, I'd remind you that none of this happens if we don't get financing. So we have these needs for humans, and if you are just strictly focusing on planet Earth, you know how most of these things are solved. But we have very different challenges when we go beyond Earth, and the biggest challenge really, I believe, is upmass. And that is talking about launching mass up from the surface of the Earth. So a lot of the mass of a spacecraft goes into just those first few minutes, lifting it off planet Earth past the atmosphere and out of Earth's gravity well. And because this takes so much power to do, anytime that you can decrease the mass of your payload, so much the better. So if you're in a lab, you may not worry about having an instrument, say a spectrophotometer that weighs, say, 50 kilograms. But if I'm going to have something like that, I need to have it maybe a half a gram or a gram. It's got to be much, much reduced in mass. And this is tied in not just with mission capabilities, but also the cost. And the number that I've used for years is it costs about $10,000 to launch a can of Coke into low Earth orbit. Now, that's not obviously not a standard unit, neither the dollars nor the can of Coke. But to sort of bring it home, I've been talking to some of the commercial providers to get payloads onto the surface of the moon. And right now, they're charging about $1 million to get a kilogram onto the surface of the moon. And it's not even clear that that $1 million will buy you communications or heat or even a robotic arm or leaving the spacecraft. It may just be, we got you to the surface of the moon. That's it. So you can see that this reduction in mass is incredibly important. Also, because we're so distant, we need to worry about the storage of materials and that they be reliable and flexible. It's sort of the ultimate field trip. I don't know how many of the people who are listening to this talk have been on field trips themselves, but you get out into the field and you forget a pipette min, or you run out of tips, or you forget your thermometer or your light meter or whatever. it's really annoying, and you're going to have to learn to be flexible. But sometimes you can cheat a little bit. You could call Amazon and get a new one, or call someone in the lab. But you can't just do that kind of thing when you're off planet. You can't cheat. So you better be able to have what you need. It better be reliable. And if for some reason you don't have what you need or it breaks, you have to be flexible. You need to have power. Once again, this becomes incredibly important off planet. And because I've been invited to talk in a lot of sustainability conferences the last year, I've suddenly realized that really what we're talking about is self-sustainability, that when we think about going off planet, this is the ultimate circular economy. So again, I know that my European colleagues who are listening to this have heard much more about the circular economy and the UN sustainability goals than we tend to talk about in the United States. But it's also a way to get at this issue. And I'll explain a little bit more about what I mean about that later on. So the technology that's gotten NASA and other space agencies where they are was the technology of the 20th century. We built big rockets, we powered steam engines, we had computers. It was all the age of physics. But look at all these problems that I've outlined for you, the needs of humans and the constraints that we have. And I believe what I have in this black box is going to be that much more powerful for the 21st century. I have a technology that's programmable. Well, so are computers, and it's modular. And once again, so are computers. So that's, you know, you're sort of on track, but it's self-replicating. And I don't know about you, but my cell phone doesn't replicate. I buy a cell phone. If I need a new one, I have to buy a new one. It doesn't make its own babies. And it's self-repairing. And now you really are well beyond any technology that the physicists have come up with. And by the way, it's got a whole lot of other really, really cool attributes that, that nothing else has. And I'm sure it's not going to surprise this audience to know what I have in this black box. And what I've done is I've just sold you life as a technology. And this is a really big, it sounds very simple, but it's a huge change in mindset. I look at my colleagues who are technologists, and they're somewhat leery of looking at biology as life as a technology. And I sort of only half joke with them. I hold up a cell phone. I say, look, your cell phone's rated for what, 2 or 3 years? And now look at my leg. That's rated for about 100 years. So which technology is actually the more reliable? It's because of this ability to repair and replicate and all these things that biology is an incredibly cool technology. In fact, this is kind of a funny, maybe summary of what life as a technology can do. And I hate to use the word biology because biology is the study of life. So let's say life. So if you think of life as a micro machine that does carbon chemistry and nanotechnology, it should just make your head feel like exploding. There's so many cool things that can be done. And if you add in synthetic biology, which I'm sure many of you know what it is, but let me just give a very simple definition as a way to get everyone on the same page. It's just the design and construction of new biological functions and systems not found in nature. And this is one that was used with the White House National Bioeconomy Blueprint. And basically, it's what the British have been using for years. And this is probably as good a summary as any way. So it's sort of an easy way to think about it. So what is the big idea? When I started this program, people asked, what's going on? What are the real concepts, particularly when I had to present this to fancy committees at NASA? And so here's the big idea. So this is going to be the rest of the talk, is me giving you examples of what I think are mind-blowing big ideas. Okay, so let's start with material science. As it turns out, I just spoke last Wednesday at the Materials Research Society meetings in Boston. And so we're all about material science. Now, we think when we talk about material science, we might think about, man-made materials, polyesters or nylon or, whatever, graphite. But the materials that we have used for literally thousands of years have been biologically produced. So here's a picture I took outside of Glasgow. And there's obviously some beautiful sheep. And clearly we use the wool, we use the leather, we even use some of the innards for sheep. There's a tree in the background. It's a little hard to see, but I made sure there was a tree there. We've certainly used wood over thousands of years. In fact, someone mentioned at the meeting last week, materials that we use are so important that actually we've named a lot of the eras of human history out of the materials we use. Think about the Stone Age, the Bronze Age, and so on. And so here we are now in the biomaterials space. And so we have these organisms that provide all sorts of great biomaterials, whether it's sheep or silkworms or trees or, cotton plants or rubber. But we can't take things like that. I mean, we might be able to take a silkworm, but the others are pretty much out of the question. But what we can do is take things like Bacillus subtilis. So this micrograph shows Bacillus subtilis. It's a common soil bacterium, and the white dots are spores. And the spores are particularly interesting because they are extremely resistant to the space environment. In fact, they have the best flight heritage of any organism I know about. They've been flown on the long duration exposure facility for nearly 6 years. That was not the intention. It just happens that they couldn't get them back down earlier. And many of the spores survived that. So you can't really beat 6 years exposed to the space environment orbiting the Earth. So why not take this form factor that is so good at living in space and put some of these capabilities in this form factor so you can now access what I like to call nature's hardware store and bring it up in a different form factor. Now, on top of that, organisms on planet Earth actually provide the interface between the raw materials on the planet, whether it's carbon dioxide, light, water, and minerals, and then the products that we use, these biomaterials. So the organisms that do this first interfacing are primary producers. Most of them today are the photosynthetic organisms. So you can think of grass and algae and other plants and lichens and so on. And then they produce sugars and other organics. So they have been able to transform these inorganic raw materials on the planet to something like a cow could eat, that then might be producing materials for us like sweaters and drugs and habitats and so on. And we have suggested that you should have a similar system off planet. I mean, this is a system that's worked very well and has evolved over and over for the last 4 billion or so years. Why not do the same thing off planet? So our vision is taking something like a photosynthetic alga or cyanobacterium that, again, is the interface between the raw materials on the planet and, say, a production facility that has maybe your Bacillus subtilis that has been engineered to produce nanocellulose or drugs or whatever. And so we can have a parallel system. So let me give you a more concrete example. And that's a project that we've been working on for a few years on habitats off planet. Now, here is a photograph from Apollo 11. I don't know everyone who's online, but I'm going to go on the assumption that I may have been the only one who actually stayed up all night and watched Neil Armstrong and Buzz Aldrin walk on the surface of the moon. And it was incredibly exciting. But look at the habitat that they had to live in for 36 hours. This lunar module was very small and cramped. And if you're the first person to be on the surface of the moon, or even probably the 50th, yeah, I could live in a little thing like that for 36 hours. But if we're going to have a sustained presence on the moon, we can't expect people to live in tiny quarters like this. They just won't be able to do much. They will be tired and cross, and you don't have room to do anything really. So this traditional approach that we've used for space exploration is to bring our habitats with us. In fact, the nominal mission for Mars is to bring a 29,000 kilogram habitat for 6 astronauts. So you hear 29,000 kilograms, that's an awful lot of mass. So is there a better way? Now, a few people have suggested things like a robotic construction of a regolith or an ice house. A regolith is just the surface material on Mars and the moon. What we have suggested is, again, turning to life. And the life form that we have turned to are the filamentous fungi. Now, what am I talking about? Well, you're probably all familiar with yeast, you know, makes beer and bread and all that. And these are unicellular fungi. But all the rest of the fungi are formed of filaments. Some of them form fruiting bodies like mushrooms. But the main part is this filamentous structure that for a mushroom is underground. And this is the fungal mycelium. You can see that micrograph I took of the hairs of a mycelium. And it turns out that if you build a mold and put this mycelium in it with something to bind, you can actually create a structure. And we've done a little bit of that in my lab, but we had the idea, gee, wouldn't it be really interesting if you could build, well, actually not build, grow a whole habitat off planet. And thus our mycotexture off planet project was formed. We have a couple of architects on it, primarily Chris Maurer, who's the principal architect at Red House Studios. We have an architect in England. We have planetary scientists. We have a really amazing team, fungal geneticists and so on. And this mycotexture is simply mycology, the study of fungi, combined with architecture, and says what they call a portmanteau, mycotexture off planet. And hopefully this movie is going to play. This is Chris doing a great job of showing what this concept's all about.
In the near future, shortly after an unmanned mission lands on Mars, a hermetically sealed bag is delivered that contains microbes and spores. A rover delivers carbon dioxide, nitrogen, and water sourced from Mars. The form fills with air and water, and the embedded algae grow. The reaction creates oxygen and biomass to fill the form.
I'm going to stop that there. But you get the idea that what we're talking about is making a biocomposite. Now think about the atmospheric pressure is very low on the moon or Mars, so the algae shouldn't have to make all that much oxygen. They provide biomass, and all this is done in an inflatable, partly because, of course, you don't have liquid water on the surface of the moon or Mars. And so you have to have a little more atmospheric pressure in order to have liquid water for the algae and the fungi to grow. But also, as I'm sure you're all aware, when you get to Mars, you have to worry about planetary protection. So what have we done so far? We're on our third year of 2 different grants of funding. And we have some prototypes we started to make. This is a prototype of the inflatable that a company called Moonprint Solutions that specializes in inflatables off planet made. And I just think this is really fun because it shows that this whole thing's real. It's obviously a time-lapse image. This is a 4 by 4 meter little prototype. And I mean, I just think that that's great. I love it when we actually can see and touch things. So how does this work? We've tried all sorts of different things. On Earth, people tend to use wood chips as the material that the fungi bind, or clippings from the garden like grass and so on. Obviously, wood chips and garden clippings are not going to be in great supply off planet, although they are probably ultimately going to be growing some plants, and there'll be inedible parts to that. And so my 2018 Stanford Brown iGEM team worked a little bit on this, and they tried a whole lot of things from my pantry, including coffee grounds and Martian and lunar regolith simulants. So what they did is they added a little nutrient broth to the simulant, and they could clearly get the fungi to grow on and bind the material, which to me is incredibly exciting, and this point's the way to go. And here's a little stool that the students made, and you can see in 2 weeks you can make a stool out of fungi that is rated for a human being. So really exciting. We've done a little work with the cyanobacterial fungal composites, and I don't want to take a lot of time on this, but for those of you who are more sort of microbiological, the disc that you see in A is a dried nostoc flagelliflora, which is a diazotrophic cyanobacterium. So what that simply means is it's a particular cyanobacterium that not only can fix carbon dioxide, but also nitrogen, which could be very handy particularly on Mars. And then the second one you see is the hydrated one that is now inoculated with Aspergillus oryzae. So it's got a fungus inoculated in it. You see it fully colonized. You can see the fungus is overgrown the cyanobacterium and it looks white. And then all the way over on the right, you see ones that were then compressed and tested for structural integrity. So now you've got this building material, which is really cool. We're trying other sorts of algae right now. Here's a picture. I'm over on the left and Chris Maurer is over on the right in Chris's studio in Cleveland. And you can see he's starting really big prototypes and playing with sand as a stand-in for the Martian and the lunar Regulus simulant. So really, really cool stuff that we're moving ahead on. Not everything we do is going to make it to Mars and the moon. I am determined to get this to the moon. But in the meantime, we also have made a connection with a restaurant in the Basque region of Spain. It's not really a restaurant. It's one of the top in the world. And it's been often nominated as the top sustainability restaurant in the world. This is a restaurant called Azurmendi. And they have some students now starting to work on nouvelle cuisine with fungi. You can see these great little balls on the right. And I believe that with a little luck, one of their students is going to spend some time with us next year. And then Chris has also designed a NASA corner at the restaurant. And you can see this has got tables and chairs and everything. I just think it'd be really cool to see how much we can do building with mycelium. Okay, so that's sort of some of the spiel on material science. Obviously, there's a lot more that we can do, from nanocellulose and spider silk. And, there are a lot of things that are out there that people are working on. We've done a little work on, many people have done even more, and then combine it with bioprinting. Huge, huge space in material science. So let's move on to another big idea, and we send digital information to make DNA. This doesn't sound like a big deal for those of you who work on molecular biology. You do this every day. You come up with sequences, you push send, your supervisor puts a credit card in or whatever, and you get the physical DNA. And you don't think twice about it. But it is a big deal. It's a really big deal, because up to, just a few decades ago, the only way DNA was made was from pre-existing DNA in a cellular form. So now you no longer have to do it. You've broken that link. And there is nothing lower mass than sending digital information. So now if you want a piece of DNA on the moon or Mars or ISS, if there is a DNA printer there, a synthesizer, you can just send the information and synthesize on site, which is huge, huge deal. Now, biology does chemistry on the Earth. Why not beyond Earth? And since I'm going off wine tasting day in California, this just simply reminds you of all the chemistry that life does. I would guess some large percentage of the Sigma catalog is also based on biologically produced materials. Now, here's another problem. We mentioned that humans need pharmaceuticals. Now, up to now, when astronauts go into space, they're very carefully tested for their health and so on. But ultimately, as they're longer duration missions, and we really can't get an astronaut back quickly to the Earth if there's a health problem, you're going to have to worry about medications. Now, a round trip to Mars, the nominal mission is about 2.5 years. So you figure about 6 months to get there, about 1.5 years until the planets realign, so that you can have as short a time in transit as possible, and then 6 months back. And so and then of course you load the spacecraft in advance. So you need to have something that's stable for, say, 3 years. Longer, preferably, if something happens wrong. But many of the new, really exciting drugs expire much more quickly. For example, biologics, these peptide-based drugs, only have a shelf life of about 6 months max, and that's under refrigeration. And so with some encouragement from Phil Williams, who is a pharmacology professor at the University of Nottingham in the UK, we've developed an astropharmacy, his name, and it's a miniaturized on-demand production system. We have a cast of 1,000. I'm pretty sure Kate Adamala may still be awake listening to this, and she's been an amazing part of this team as well, and I think is the only Polish colleague I'm going to show today besides Tomasz. So that's my Polish, and I may have some Polish ancestry, but let's just leave it at that. Anyway, so the astropharmacy, the idea is to do something that's radically different from what pharmaceutical companies do. Pharmaceutical companies want to make money. So they want a drug that they can make, you know, literally hundreds of thousands of samples that they, preferably chronic, they can make some money out of. Whereas we have exactly the opposite problem in space, where we have a very limited number of people who need it, but when they need it now. And so our concept is to pre-program Bacillus subtilis, or if we're able to have a more stable cell-free system, you combine that with cell-free synthesis and then just synthesize on demand. And what we're trying to do is get ourselves down to a 24-hour time period with synthesis and then purification and microfluidics. And so very exciting project. We're getting ready to apply for a phase 2 on that. Now, remember I said at the beginning that life is really good at carbon chemistry, which we've been talking about, but it's also really good at nanotechnology. Now, think about it. If you're making a polymer, for example, in the lab, you're lucky if you can be accurate within 10 or 20 monomers or 1,000, maybe even 100 monomers. Or if you're trying to place things in a normal sort of nanotechnology approach, you've got a fancy clean room and bunny suits and all sorts of things. But in the meantime, life does that every day without a clean room, without even thinking about it, because we have to have that kind of precision to be alive. I mean, think about it. You get one base pair wrong, you can end up with sickle cell anemia, or a whole lot worse, you could end up being dead. And so what if we could take advantage of this power of precision that life does without even a clean room? So Simon Vecchione, that you see over there, was the captain of our 2013 iGEM team. He was a senior at Brown at the time, interested in physics. And he came to me with the idea of building a radio transmitter using life. And I don't know about the rest of you, but I don't think I could build a radio transmitter even if I had a kit to do it. And by the way, for those of you who are interested in SETI, the way the SETI people define intelligence is the ability to build a radio transmitter. So I always joke to them that I am unintelligent in life and proud of it. But Simon wanted to do that, and it seemed awfully ambitious to me for the summer. And so we talked him down to a project that he came up with, and that was to build wires. So you're all familiar with DNA. It turns out that if you have two cytosines, rather than a cytosine guanine pair, you have a cytosine-cytosine mismatch. It turns out that a silver atom can be incorporated. And by the way, if you have a thymine-thymine, you can incorporate mercury. Now, if you just know this and you think, well, silver, you can form a wire. And think about it. DNA is linear like a wire. Silver is conductive. And so what he did is form linear wires with us. For his PhD, he then got into two-dimensional systems, and now in his postdoc, he's working on three-dimensional and all sorts of interesting applications. Here's an NMR, a cartoon from an NMR that he took fairly recently, and you can see in the end, it turns out that there are 3 silver atoms that are incorporated every cytosine-cytosine mismatch, not one. And so again, think about it. You can design a sequence where you know where every silver atom is. You could go every other one. You could go 3 cytosines and then 1 GC clamp. Or you could I think he's done 20 or so in a row with clamps at the end. So, I mean, you can do all sorts of really cool things with precision without a clean room, without bunny suits, taking advantage of life. Now, another thing that life does is it converts chemical energy into electrical energy. And this is a really cool idea. It's not just the electric eel. All of you are doing that as well. This is what life does. And so it's possible we could harness this power of the conversion of chemical to electrical energy. That's basically what a battery does. So again, let's think of another big idea. And this is one I alluded to a little bit with the wires, and that's the idea of using life to extract metals. So biomining is incredibly important. Think about it. Metals are very massive. We need metals for our technology. We also need it for life itself. So here is the traditional way to do mining. This is a mine that I visited outside of Las Vegas that you can see. It's a very big operation with big, bulky, heavy, expensive equipment. and facilities. Clearly, build something like this off planet would take an awful lot of missions, and I can't imagine it really would be worth it. And here are the major elements that we need for space exploration. We need lithium for batteries, we need aluminum, we need nickel, palladium, silver, gold, also carbon, oxygen, silicon. And here are some of the major elements for life, and you can immediately see overlap. And as it turns out that these are just the major elements for life, that actually many of the elements in the periodic table are bound by some peptide or some biological compound or another. So we have a really nice database that we've accumulated in my lab of both naturally and synthetically occurring peptides to bind most of the periodic table. And so taking that information, we had proposed to NASA that we could recycle integrated circuits. Now, an integrated circuit doesn't have much mass, so you think, well, it's no big deal to take more up. But the problem is they're not completely radiation resistant, so eventually they're going to go bad. So what if you could take this integrated circuit, grind it up, and take a peptide that could pull out, say, the copper or some of these other metals. And people ask me, which metals does life use? And I say, look at the back of a vitamin bottle, and that'll give you a head start for just humans. So you've ground up this integrated circuit, you combine it with a metal binding peptide, and then you combine this with something like a plasma printer, which our colleague Ram has developed. And so he uses this peptide that has copper bound as an ink and was able to reprint an interconnect. And what's really cool about the plasma printer is you then vaporize the peptides, so we didn't even have to purify them. And once we thought of that, and then we combined it with the fungal mycelial project, which you remember from the habitats, we have a nice paper out on that, and we have a patent pending, which they promise they're going to look at. It's been the US Patent Office since October 2020. And the idea is that we've been able to take fungal mycelia, create a filter, and then pull out metals that way. So think about it. You've got a substrate for a filter that is really cheap, that can be grown into any form and then bind any metal. So we're extremely excited about it. Now, when you have this biological vision, as you're well aware, off planet, the environment's very different. So if you're having to heat up a bioreactor in order to make your pharmaceuticals, it would be great if you could save a little power and maybe have the optimum for your bacillus be a little bit lower. And so this is where we've looked a little bit at a project that the iGEMers in 2012 called the Hell Cell. It's basically pushing the environmental boundaries for life. But you could not only do that, you could go one step further and maybe create life de novo, Life 2.0. And I know there are European groups. We have a group that's NSF funded called the Build-A-Cell Consortium. And again, Kate Adamala has been running a fabulous series on Mondays. It's morning for me. It would be evening for people in Europe, of people giving talks on various aspects as we go towards trying to create a new cell, whether it's a bottom-up approach from different components, or taking a top-down where you take an existing cell and you minimize it. By the way, Kate does videotape those, and so you can always go on the Build-A-Cell website and see some of these talks. And so I say every week I am just astounded by the quality of research that's going on around the world. So I really recommend that. Now, people say to me, these are all, great visionary ideas, but are they actually going to work in space? And so we were really lucky a few years ago to be invited to be a secondary payload on a German satellite mission called EuCROPIS, Euglena crops in space. And their idea was to have a system with Euglena, which is a beautiful little alga-photosynthetic protozoan. So it's a protist. And growing tomato plants and recycling artificial urine. And just to cut to the chase, they had telecommunication problems, so there are no results from theirs, but plenty from ours. One of the reasons I was interested in EuCROPIS is not just because people don't offer me a satellite ride every day, but because the concept was to test some of these systems at different gravity regimes. So it started off at microgravity, not spinning, and then it spun up to about 1/6 Earth's gravity, so that's lunar gravity, and then spun up to 1/3 Earth's gravity, so that would be mimicking Martian gravity. So we had a chance to do this several times. So we called this payload the PowerCell payload, and the idea of cast of 1,000, who helped, and I don't mean to minimize that because we had a lot of really fabulous people helping, but I'm also cognizant of the time. So the idea of PowerCell was, again, as I said at the beginning of the talk, this idea of having an organism that's the interface between the raw materials on the planet and feeding something like a Bacillus subtilis, which would be our production organism. And so we had microfluidics cards. All these had to be done in 48 well microfluidics cards. The only thing I had is some fluid flow, anything we could pack in advance, and every little well had its own little tiny spectrophotometer. And you can do a lot of science with that. So we thought, well, we've got PowerCell, but then we got 48 wells. So even with replicates, why not try a transformation in space? And why not try the strength of promoters? And to me, I'm sort of agnostic on the results, because if it turns out that there's a big difference in gravity, we need to start testing space quickly so that we can allow for that. But if not, that's great, because then we don't have to, and it's a lot cheaper to test these things in the lab. So let me cut to the chase. The PowerCell worked really well. The transformation worked at the beginning. It turns out, word to the wise, you cannot leave sugars in LB on the counter for years at a time. They will eventually undergo a Maillard reaction and caramelize and become inedible. And the promoters, I'm going to give that sort of an empty, because there was some effect, but we expected one promoter to work much better than the other, and it didn't. So what I've given you is a lot of ideas for how we can use synthetic biology for this idea of astrobiology, of going beyond planet Earth into the future. But there are other parts to NASA that, besides the exploration, there's the aeronautics. That's the first A in NASA, National Aeronautics and Space Administration. We believe there's a lot of future there. Some people are working on that commercially. We may get into that space eventually. And then, of course, there is science, which is what many of you are interested in. And we believe that synthetic biology can help answer questions, the origin of life, the limits to life, and so on, because it allows us to ask the what if questions. What if something had happened differently? And so we have wondered this question, are we alone? And I'm going to argue that synthetic biology will help. And this would normally be a full other talk, and maybe someday I can give this talk to the Astrobiology Society of Poland. But normally what you would do is ask, what are you looking for? What do we have in terms of the diversity of life on Earth to give us concrete examples of where life could potentially live? Then go out into space and map where these extremophiles might be able to live in other habitable bodies. But I'm saying there's actually a fourth step. What if you find locations that are beyond the capability of extant naturally evolved extremophiles? Why not turn to synthetic biology? And this goes right back to this health cell project. So we can really do this two different ways. The question of using this to help the technology off planet, but also to look for life. And what the iGEMers did, this is the 2012 team, is look at specific genes and operons to try to push E. coli to become more desiccation resistant, radiation resistant, pH, heat, and cold. Many of these worked. Here's an example with the desiccation resistant. The blue bar shows the control E. coli. And as you can see with the Trehalose biosynthetic operon, there's a bit more desiccation resistance, not surprisingly glycine betaine, and somewhat surprisingly manganese transporter actually improved the desiccation resistance enormously. So this is a fabulous approach. But there are others as well, including looking at functional metagenomic screening. And what you see here is a picture of a relatively new postdoc in my lab, Garrett Roberts-Kingman, who is taking up the mantle of the Health Cell Project, and I couldn't be happier. You might imagine that he's evolved some incredibly super GFP strain there that's making everything glow. But I will have to be honest that this was a French film crew with a green light, but it's a great picture. And so I'm so excited that Garrett's working on this, and I believe we'll have more people even come in the spring. So let me close with a few final thoughts. I mentioned at the beginning that I believe that going off planet is actually the ultimate biosustainability argument. So on planet Earth, we make things, we use them, and we recycle them. That's the biological cycle. Our technology cycle is really not a cycle. It's a one-way path right now. We take, we make, we use, we dispose, we pollute. We need to close this loop. And I've showed you a couple of ways with, for example, biomining, we could do this. When we go to some place like ISS, we're very good at recycling some things. The line you may have heard is yesterday's coffee is tomorrow's coffee. I don't think I need to explain that to anyone. But it's very expensive to get things up there. And I've discovered that not everything's recycled. In fact, dirty laundry is sent overboard and deorbited. It's burned up in the atmosphere. This is not sustainable. Now, when you go off planet, we don't know if there are biological cycles elsewhere. We know they're not on the moon. Who knows if there are some on Mars or in the, at least in the distant past. But we are going to start semi-biological cycles and certainly technology cycles off planet using in situ resource utilization. We've got the radiation, gases, energy, minerals, metals, water, spent spacecraft. All these are raw materials that we could then use, hopefully make something, use it, and then hopefully recycle it and get a real cycle going that will be really incredible for off planet. And so again, we have these constraints when we go off planet, but I believe the constraints are not depressing. They actually make us be more creative, and they're forcing these game-changing solutions. And so I'll finish the sentence I was going to say in a slide ago, and that is that I firmly believe that these technologies that we're developing will ultimately feed back into revolutionary technologies to help solve the problems on planet Earth and, of course, beyond. And with that, ad astra, which means to the stars. And thank you to the many, many people who have helped or been through my lab, or both, during the last, I guess it's about 10 or 15 years. Thank you very much.
Thank you very much, Lynn. That was absolutely fascinating and brilliant talk. You get a lot of comments and a lot of appreciations from our audience. I have a question for you. Before I go to the questions from chat, my question would be, what are the biggest challenges with the use of biologicals in space? Like, first things that come to my mind, there could be mutations, infections. So what else could happen wrong, and how do we prevent that or mitigate?
Actually, whoever asked the question's more advanced than I am, because I think the first problem I worry about is making sure that everything isn't dead when we get there. That it's alive and it's functional and it does what we need it to do, because we're going into environments where life does not normally thrive. So I'm more worried to starting with that. In terms of infections and so on, obviously we're going for things that would have a very low infectivity rate. Hopefully we will be focusing on not having them mutate, except when we're trying to select for mutations. I mean, I do think that we're asking organisms to live in environments that they did not evolve for. And some things we can mimic on planet Earth, like temperature and so on. Others, like gravity, we can mimic it, but the mimics aren't very good. And so you're much better to do directed evolution in the actual environment that they will ultimately need to be functioning in. But I would suspect, I mean, the way I would want to go is that something like a Bacillus subtilis spore is incredibly stable. I mentioned not just with space radiation, but, dried, they will last for years. And so to me, that's, and they will literally dry on a piece of paper. So their mass is virtually nothing. So what I would do, and this is what I'm suggesting for the astropharmacy, is you can make thousands of different engineered Bacillus subtilis cells, dry them down on little tiny pieces of paper, and you have them maybe in the size of something like a cell phone, and you can just keep pulling out fresh ones. You don't need to worry about it going bad. So I think that that's some of the answer. There are all the usual sorts of tricks with kill switches and antibiotic resistance and so on, which you want to avoid the antibiotic resistance because you then would have to keep making the antibiotics. But they're the clever sort of tricks for that sort of thing as well. But I think that I would feel better having fresh inocula that I could always turn to if I needed to, just like we turn to NEB or any other company to give us a fresh sample if we need to.
Very interesting, and yeah, brilliant answer. So either we evolve the microorganism there to already be adjusted, or we just take as many copies as we want, because when they dry out, they don't mutate that much. So yeah, smart approach. I have another question.
I need to be complete. Yeah. I just need to be completely honest that we have a proposal that's in the final review to do exactly that, to do this evolution off planet.
Very interesting. Yes, more people congratulate on the talk, and the question from Majana Kshak is, it's a very interesting talk. My question might be side off. Are some microbial organisms considered to candidate as a polyfunctional in the needs, like biomining and electricity generation at the same time?
Ooh, I hadn't thought about that, but that's a really cool question. Let me think about it. That's a great idea. Whoever thought of that, that's great.
Thanks. Then another question. I hope another question that we got is, Pedro is wondering about the applications of genome editing like CRISPR on the organisms in these applications.
Yeah, no, absolutely. Genome editing and yes, absolutely. That's the kind of thing that we do. What I wanted to do for this talk is to inspire people rather than going into the actual details of how we're doing all these things technically. But yes, of course, you're right.
Yeah, I remember when we were working on the microarchitecture, and there were ideas to modify, for example, bacteria that would adhere to the fungus, giving it even additional functionalities. We're checking like colors, right, and things like that, just as a principle. But there's a lot of modification.
Absolutely.
And you don't even have to
I still want to do that. In fact, there's someone in the Netherlands who's done something relatively straightforward in that he's knocked out the gene in the particular filamentous fungus that he is working on that allows it to form a fruiting body, in other words, a mushroom, and he can get much more uniform mycotexture. And so clearly we need to do that as well. So, I mean, even something simple like that can make a big difference.
I have a question, like, which of these technologies you think are priorities, one for the moon and the other one for Mars?
So our priority right now is certainly the moon. We've got, the Artemis mission. So that I'm required to say Artemis like 5 times every talk. It's, I do apologize for the sound in the background. It's my husband's birthday today and the phone calls are starting.
Happy birthday.
Yeah. Anyway, so the moon is the priority now, but in some ways Mars is going to be easier because there's a little more gravity, there's some more raw materials. And so I'm excited to think in terms of Mars, but certainly again, the moon is our current priority.
Okay, we have one last question before we finish the session. So Marek is saying that we have found bacteria that can eat metal. Maybe in some point in time we would be able to engineer bacteria with low cost and extract metals from ones, despite it is very energy consuming process. Yeah.
Right. So this is certainly something that humans have taken advantage of in terms of biomining, of precipitating metals or, ingesting them and forming nanoparticles. Certainly the extraction been for literally thousands of years. I know 2 people at the University of Edinburgh who have been very interested in that. Louise Horsfall is making nanoparticles with bacteria that do ingest certain metals. And former postdoc, Charles Cockell, who's professor of astrobiology at the University of Edinburgh, actually flew something on ISS, and I think it was the Stone mission, but I could be wrong, where he was testing some of these extraction techniques. So there certainly are organisms, in fact, there are fungi that naturally bind metals. Our attempt is to make a tunable platform where we could bind anything you wanted. So I think some kind of combination's good. Not every metal could be ingested and formed into a nanoparticle because many of them are toxic. And so our focus has been to try to keep them outside of the organism so we literally could do anything. So again, you're absolutely right. There are other uses for that. I believe firmly that the nanoparticles can be of use for other things. Again, I would encourage you to take a look at Louise Horsfall's work in that area. I know right now she's doing some work with actual I think they're lithium-ion batteries that are spent, which I think is really exciting to start to get your hands on real materials that you want to recycle that are really nasty. And if you can do it biologically, how cool would that be?
Thank you, Lynn, for your answers. Unfortunately, we don't have more time for questions. I have one more advertisement to make, but also I want to let you know that a lot of people are saying happy birthday to Rocco. So please forward that.
Thank you. And thank you very much. And I would like to know when I'm going to get an invitation to Poland, because I've never been there.
Absolutely. And yes, we would be totally delighted to host you here. And if you could give us another lectures, that would be wonderful. Thank you so much. Enjoy your day.
Of course.
Say happy birthday to Rocco.
Thank you. And thank you. I certainly will. And thank you to everyone for staying on this long on a Sunday night during the World Cup.
Appreciate it.